just briefly to do the histology. And your quiz is going to be back to where we started on Wednesday, 192, to the end of liver histology. That's quite a bit of material. And so your quiz will have, like we did last time, your quiz will have 7 questions. All right? We have a quiz every day from here on out. There's only 3 more days. We have 3 more quizzes. So quiz 9 will be on the digestive system on Wednesday. Quiz 10 will be on the urinary system on Monday. And then quiz 11, which is your optional quiz that you may use to drop in the lowest quiz, will be on the reproductive system. That will be on the lab for a regular lab. So on page 199, we talked about the plicae circularis this morning in lecture. And the plicae circularis are referring to the visible circular folds that you can see in the small intestine. Those are not shown on this page. Letter A on page 198, 199 is demonstrating the plica circularis, and it's actually showing a cut, through the circular fold. So this is a fold of mucosa. Remember how I diagrammed it in lecture this morning? Each of these extensions coming out here is an individual villus. And so this is showing a single villus with its covering of simple columnar epithelia and a lacteal inside it, similar to what we saw in those histology models. Although this is smaller, one of these is shown here, or here, or here. So this is showing a plica circularis, cut in a cross-section, or coronal section. So on our 3-dimensional model, that's shown here. Remember, it means circular folds. So if I say identify this fold, I'm looking for plica circularis. Okay. This structure right here is the duodenum. Here's the superior mesenteric and the artery and vein, but it's not extended far enough to show you the right angle bend where it becomes intraperitoneal as the jejunum. So those are shown on additional diagrams or models. This older one back here shows a real good, not that one, older, white one, here, this one. So if you look, here's the duodenum, and then right here just under the stomach, you can see the duodenum makes a right angle bend, kind of a knob-like structure. That's the beginning of the jejunum. Okay, and that's where I would tag it. It's just distal to the duodenum, and you can also see it on these charts. There's another chart over there to the side. So again, here's the duodenum with the pyloric sphincter, plicae circularis, coming around superior mesenteric vein and artery. Here's that knob again. And so this is retroperitoneal, and it becomes intraperitoneal at that point. So right in this area is where I would tag jejunum. And then ileum, remember, is the most distal portion. So I would tag ileum somewhere here where you can see it actually attaching to the cecum and ascending colon, because it's the most distal region. All the coils in the middle, I can't figure out which is which, so I wouldn't ask you to be specific as to jejunum or ileum. So on the diagram, letter S is showing the duodenum. Letter Q, it's really hard to see, but letter Q, if you draw the line all the way up, is supposed to be the jejunum. And then R is showing the distal end of the ileum. And T, down at the bottom of page 199, is the word mesentery. Would you remember what I asked you to identify it as in lecture? Mesentery proper. Okay? And you can see that here. So here's the jejunum, and here's the mesentery proper, enfolding the jejunum, which is intraperitoneal. You can also see mesentery proper on this chart. down in the last lab. So here's a diagram similar to what I drew on the board on Wednesday. There would be a cut through the stomach and the colon, and here's the loops of small intestine. So each of these right here is mesentery proper. Okay, back to the intestinal wall model that we looked at just briefly for the template portion. So remember, B was mucosa, and the surface epithelium of mucosa is the simple columnar, and that is letter O, containing goblet cells, which are P. And the epithelium lies over a loose, irregular connective tissue or lamina propria, interchangeable terms for our mucous membrane, which is W. It's all the gray that you see in here. Down here, the gray is dense irregular. This is not lamina propria. All right, but the loose irregular up here would be lamina propria. This would be a villus. So that would be one of the structures indicated by F. And the intestinal gland is the loop that goes down into the lamina propria, and that would be letter I. And then our muscularis mucosa is letter G. Submucosa is C, and other than the lymphatic nodule, remember I said to change node to nodule or follicle, letter H. There's really nothing else remarkable about the submucosa, and I don't really tag arteries and veins and nerves on there, it's just they're found there. And then letter D is the muscularis externa. For those of you that weren't here Wednesday, I asked you to add the word externa to muscularis and add the word inner to circular, just deep to the submucosa, and outer to longitudinal. So M would be the inner circular, and N would be the outer longitudinal layer. And then finally we have the serosa, or visceral peritoneum. Use either term for letter E. The lacteal, would be the green, single blind lymphatic capillary, which is letter letter U, in the diagram to the left of the villus. All right, looking at the large intestine. So I'm going to go through the regions of the large intestine and come back and look more at the detail. So we have the ileum entering, attaching to the large intestine at the point where the cecum ends. And so C is our vermiform appendix. You can just call it appendix. You don't need to include the word vermiform. B would be the cecum. And A is the ileocecal valve or sphincter. Doesn't have as much smooth muscle as the rest of our sphincters, so that's why it's sometimes referred to as a valve. And then F would be the ascending colon on the right side. It makes a bend or flexure, and so either right colic or hepatic flexure, since it's on the liver side, would be letter G. H would be the transverse colon. And on the left side, letter I, we would have a left colic or splenic flexure. And then J is the descending. K would be the double S-shaped curve for the sigmoid colon. You're not seeing much of the rectum. It's kind of covered up here, but you can see more of it on the chart. And then this last bit right here would be the anal canal. So M is showing only the distal end of the anal canal. It actually goes, the narrow portion would be the anal canal, and the anus is the opening. So N would be the anus, which is the opening, kind of like the external nares to the vestibule of the nose. In the lower right, the lining, the smooth muscle wall of the anal canal is shown as O, and that thickens to form the internal anal sphincter right along the edge here. Okay, look at the chart. And so the bottom, it looks like it's part of the wall. It's a smooth muscle part of the wall. And then outside we have skeletal muscle. So this thickened portion P would be the external anal sphincter. Okay. We can look on the diagram. O is internal and P is external. All right, now, the transverse colon and the sigmoid colon are intraperitoneal, which means they have a mesentery. And so this is the mesentery or the mesocolon. Go ahead and add the word transverse to mesocolon because that's the only one I'm going to tag. It doesn't have a letter. It's called transverse mesentery in the diagram to your left. However, you can diagram it in kind of the way I've drawn these lines right here. And that's what's suspending the transverse colon. So see how those vertical lines kind of give you the idea of the transverse mesocolon. The mesentery of the colon. So these lines right here. And that's what this is again, right here. Okay, the large intestinal wall, I'm not sure why it's there because we don't tag it anywhere, but moving on. So page 202 and 203 are the liver and the pancreas. So if we look at our large model of the liver, remember it's kind of weird because the stand is actually on the anterior surface. Okay, so the right lobe is labeled P on the lower left-hand diagram. The left lobe is S, so that's the largest and second largest. And then if we look up here, remember I said that the inferior vena cava is posterior. Inferior vena cava is T. There's no word for that, but you can write it in if you can't recognize it. And so the most posterior lobe is the caudate lobe. So the lobe right next to the inferior vena cava, letter Q, is going to be the caudate lobe. Caudate meaning tail. Then here is the gallbladder, and right next to the gallbladder is the quadrate lobe, and that would be letter R. Now coming into the liver, we have the portal vein. That's a larger diameter than the hepatic artery. And so portal vein on the lower left is letter V, and hepatic artery is letter X. Now, remember the umbilical vein from the fetus comes into the liver, so it's going to enter the liver very close to where the portal vein and hepatic artery do. So letter V is the ligamentum teres, which is our collapsed umbilical ligament. Now it's traveling in the falciform ligament, which is shown as A, but I would prefer to move it down a little bit better. And when I move this chart down, this shows the falciform ligament, ligamentum teres, probably the best of any area we have. So here's the right lobe and the left lobe of the liver. Here's the umbilicus. So traveling up to the liver would have been the fetal umbilical vein. 35 here, now the ligamentum teres or round ligament, same thing. And this would be the falciform ligament right here. Okay. It's also shown here, this is parietal peritoneum. It forms a fold, which is the falciform ligament. And inside that, kind of a reverse, so this would be the falciform ligament. This would be anterior abdominal wall. And headed towards the liver is the collapsed umbilical vein. So that's the ligamentum teres. So on this liver model, you can see ligamentum teres here. Falciform ligament would be like here, but it's been cut very close to the liver, so it's difficult to see on this surface. Now, what was the fetal structure carrying blood away from the liver to the inferior vena cava? Ductus venosus. So here we have this white band going into the inferior vena cava, and that's ligamentum venosum. Okay, it's not on here, but we talked about it with the fetal structure, so you can see that there. That might be one of the structures from a previous unit we have again on the liver. All right, the upper left-hand diagram is showing the pathway of the bile ducts once they join together and leave the liver. So C is showing the right side. So C would be the right hepatic duct. D is coming from the left. That would be the left hepatic duct. And the two join together to form E, which is the common hepatic duct. K is the gallbladder. The gallbladder doesn't make bile. The liver does. The gallbladder just stores it. Okay, so if you have a high lipid content, you're having, sundaes, gone over to Gunter's and had a huge sundae, ice cream sundae, and the other 5 people in your group couldn't finish theirs, so you ate theirs as well. You'd have a lot of lipid in your diet, so the gallbladder under nerve and hormonal influence will contract and squeeze extra bile into the intestines, but it doesn't actually make it. So the duct connecting the gallbladder is F, that's the cystic duct. The common hepatic duct, which is E, and the cystic duct, which is F, join together to form the longest duct, which is the common bile duct. That's been cut here, but it's letter G on your diagram. And it's going to go from the liver down through the pancreas into the small intestine. So over here are a couple of diagrams that show that. So here's the right and left hepatic, common hepatic and cystic, and then all of this is common bile. Okay? So common bile passes behind the duodenum, passes through the pancreas, and joins up with the pancreatic duct that we'll talk about in just a moment. All right, so pancreas we did not talk about in lecture, so I've diagrammed it on the board. Here is the duodenum, forming the C shape, and tucking its head into the curve of that C shape is the pancreas. It's very wide, and then it tapers into a tail. Embryologically, the pancreas starts out as 2 outpouches, 2 outgrowths from the digestive gut. The right side rotates over and fuses with the left. Now remember, the pancreas is both endocrine, it makes insulin and glucagon hormones, but it's also exocrine, which means it needs a duct. And so here's the head, here's the body, here's the tail, and on our model, you can see that this is a weird model. It's showing a little piece of the liver, but here's the duodenum. So here is the head, here is the body, and here is the tail tucked up against the spleen. Passing from tail to the head is the duct that's collecting the bicarbonate ions and the enzymes of the pancreas and taking them to the duodenum. So this is called the main pancreatic duct. It makes a little dog leg down, whereas the accessory duct is skinny and goes almost straight. Okay? So main pancreatic duct, accessory duct, and then here's the common bile duct, formed by the union of cystic and common hepatic, passes behind the duodenum, passes through the pancreas, and then the common bile duct will join with the main pancreatic duct and form a common pathway known as the hepatopancreatic ocula. Most of that is in the wall of the duodenum. You won't see it in your models. You can see a little bit of it right here, but the best site is on this chart. In your diagram, the I would be the ocula. J is the major duodenal papilla. I labeled it in lecture, but it's not on your list. So again, this is a really complicated chart. But here would be the gallbladder. Here's the cystic duct and the common hepatic going behind the duodenum. Here you can see it passing through the pancreas. So right here, number 50, is the ocula. Okay? So this dilated region where both pancreatic and bile flow together within the wall of the small intestine. So it's this kind of oval shape right here. But it's the best of all of our, you won't see it in the models, because none of our models have it opened up to that area.
What is that called again?
Pardon?
What is that called again?
It's called the hepatopancreatic ocula. or ampulla of Vater, V-A-T-E-R, if that's a little bit shorter for you. It's, again, an eponym used for a person's individual name. So in your lab manual, finishing up here with that page, the body of the pancreas is L. I'm sorry, the head of the pancreas is L. The body is M. The tail would be N. The main pancreatic duct is H. And over on the right-hand side, the skinny little one is the accessory letter O. And ampulla would be I. Again, J is the major duodenal papilla, it's just the opening of the ampulla. All right, finally, liver histology on page 205. And that's referring to this model of probably 8 to 12 pounds, even heavier than our intestinal histology, so please don't, you can carry it back to your table, but don't drop it on your foot. This I never tag, but it is useful in understanding what you're looking at for this one. So each of these this is showing 3 liver lobules, right? They're 6-sided. A wedge has been cut out, and passing through the center is a vessel known as the central vein. So this diagram, this model over here, is essentially showing a segment going from the central vein to the outer corner, one of these 6 corners. Coming in from the liver, we had a portal vein, hepatic artery, and going out, the common bile duct. That's our portal triad. And so we see branches of each of those 3 structures at the 6 corners of a lobule. So let me go through this model with you. So again, remember I said to think of a bicycle wheel? And the sinusoids would be like the spokes. So if I took this and swung it around 360 degrees, we would have one lobule with the central vein in the center. And at each of the 6 corners, we would have these 3 major structures. The blue is showing a branch of the portal vein, the red a branch of the hepatic artery. When you label them, I'm not going to be picky about including the word branch. You can just write portal vein if you want to. And so those come together, and within the lobule join to form a series of large capillaries. A large dilated capillary. A capillary normally is just slightly larger than a red blood cell, and here you could fit 3 or 5 of them. So they're dilated capillaries, and they flow by the liver cells, the hepatocytes. Every hepatocyte faces a sinusoid. It might have a back of a hepatocyte behind it, but that one would also face a sinusoid. And so they are able to take out of the blood, remember this is combined nutrient-rich venous and hepatic artery blood, they're able to take out glucose and store it as glycogen. So that if it's been 4 hours since you ate, that glycogen can be reformed back to glucose and added back to your blood. It takes out amino acids and makes most of the proteins we have in our blood, transporting iron and providing clotting proteins and so on. So blood is flowing in this direction to the central veins by the liver cells, which are known as hepatocytes. The blood in the central veins then all comes together and form the hepatic veins, which go into the inferior vena cava. Now, where the 2 liver cells back up to each other, there's a gap, kind of like if you were to stand up to someone where your lumbar region is, you can put your hand back there. So you put a whole bunch of people lined up, and that creates a channel. And that's what these green lines are. They're called canaliculi, like we saw in the bone. And the liver cells are making bile, which are breakdown products of red blood cells and bile salts. And that helps to keep lipids emulsified, small droplets. And the bile goes in the opposite direction to a bile duct. And then the bile ducts go together to form the right and left hepatic ducts and so on. So the bile is going this direction, blood is going this direction. All right? So now let's look at your diagram on your lab manual. Everything that's in your lab manual you can see on there except for the Kupffer cells. All right? So that's a lecture concept. I can't tag it in lab. So a lobule is shown by letter J. And the lobule has with it the formed of the liver cells, the hepatocytes, which are letter G. The Kupffer cells, which are letter H, those are in the sinusoids. And then we have a portal or hepatic triad, letter A. However, that's blown up over here, and it's made of 3 vessels. So B would be the portal vein. The hepatic artery would be D, and those are both lined with simple squamous epithelia, joining together to form the sinusoid, which is letter F. And then the third part of the portal triad would be the bile duct, which is letter C, and that's simple cuboidal epithelia, so it's a little easier to recognize. The canaliculi that drain into the bile duct are letter E. The Kupffer cell is letter H, and again our model doesn't have that. Central vein is letter I. Digestive. So it's a bit of material. They're each distinctive from each other. They're not easily confused with other things like the blood vessels are, but it's a lot of different things. So hence the 7 questions for your quiz on Wednesday. Again, feel free to use any of the models up here or to take them back to your tables. To a couple of them, I use one of my pens as, say, the ascending colon. Oh, let's make it the transverse colon. This is peritoneum, okay? So when I suspend the transverse colon, this would be what kind of peritoneum? Visceral peritoneum, where it's attached to the organ, okay? This double layer of peritoneum suspending it would be a mesentery, which if it's the transverse colon would be my transverse mesocolon, the same as this structure right here. All right, well that was, now what about the ascending colon? Here's the ascending colon. Now it's only on the anterior and lateral aspects of the colon. The rest of it is now attached to the body wall. So now that's retroperitoneal, and this is parietal peritoneum, where it's covering the quadratus lumborum or the inferior aspect of the diaphragm. And the ascending colon would be retroperitoneal. Okay? So parietal peritoneum, I won't, it's arguable whether it's still attached in the organ, so essentially that should still be visceral peritoneum where it's attached to the organ, but I won't ask you that because it's really a continuation of the parietal peritoneum in essence. But if it's a peritoneum attached to the body wall, it's going to be parietal peritoneum. On this diagram up here, here's the rectus abdominis. So the peritoneum lining that deep surface is going to be parietal peritoneum. Where it's attached to the liver, or to the stomach, or the liver, is going to be visceral peritoneum. Here, where it's covering the kidneys, again, it's parietal peritoneum, and the kidneys and aorta and inferior vena cava are all retroperitoneal. So that's giving you trouble. That's a good diagram to come and look at. Okay. Anybody come in and didn't get a copy of their, not a copy, but their quiz or lab exam back?
Oh, the